The overall goal of this procedure is to image intracellular calcium signals in striatal astrocytes from adult mice using genetically encoded calcium indicators such as cyto-GCAMP3. This is accomplished by first injecting the virus into the mouse striatum. Then the mouse is allowed to recover for 2 to 3 weeks following surgery. Next, the mouse is sacrificed and acute brain slices are prepared. The final step is to perform confocal calcium imaging of the brain slices. Ultimately, immunohistochemistry is used to show that the cyto-GCAMP3 is expressed only in astrocytes but not neurons, and that the virus injection did not cause overt astrocyte reactivity.
The main advantage of this technique over existing methods like transgenic or knocking mice is that it allows robust expression of target protein and a rapid and flexible experimental implementation. This method can help answer key questions in the astrocyte calcium signaling field, such as how to reliably measure astrocyte calcium signals in the branches and branchlets of striatal astrocytes.
Prepare for the virus injection by autoclaving the micropipettes and surgical instruments. Just before surgery, retrieve the prepared AAV 5G-CAMP3 viral aliquots from the -80°C freezer and store them on ice. The next step is to fill the